Sorry, my volume was real high. It's yelling at me. Um, okay, so in that last video, there was kind of a lot to digest, right? A lot of vocabulary, a lot of process, okay? An important thing to realize is that you need to be really clear on all of that before you continue, because if you're not comfortable with setting up a Punnett square, right? If you're not comfortable with doing sort of basic, straightforward genetics problems where you would use a Punnett square, okay, then you're going to get into, it's going to get more confusing if you watch the rest of these videos, okay? So what we're going to be tackling next are things that are kind of beyond what Mendel was able to figure out. Actually, we're going to cover one thing that Mendel did know, and then we're going to, go from there. So one of the things that I want you to realize is I want you to really kind of like internalize the idea that Mendelian inheritance, as we call it, is really, it only applies to certain characteristics, right? To certain traits. So it's not like you can, do a Punnett square for every different aspect of your genetics, right? It doesn't work that way. And so that's what we're going to be exploring moving forward. Okay. But all of the stuff in the last video is kind of simple and straightforward things, relatively speaking. I know. Okay. And so make sure you're comfortable with that. And like I said, I would strongly recommend that you come to Zooms if you need some help. Okay. So where are we going from here? So let's go to things sort of beyond that first step. So one of them is, let's talk about something called a test cross. Okay. So first of all, why would you need to do a test cross? And then second of all, how would you do it? Okay. So this example, we're back to our beautiful Labrador retrievers, are back to our labs, right? And so let's say that you had a female who had a black coat. Okay. Now, that's all you know about her. Okay. And remember, this is just monohybrid again, right? So we're back to just worrying about one gene, the coat color gene. Okay. So we have a female that has a black coat, but we don't know. So we know that she's, she has the black coat phenotype, but we don't know her genotype, right? She could be homozygous dominant, or she could be heterozygous. And you can't tell that by looking at her, because remember, if she's heterozygous, because that black allele is dominant over the chocolate allele, it's going to cover it up. Okay. So you can't tell just by looking at her. So how do we deal with a situation like this? Well, one possible solution to figure it out is to do something called a test cross. Okay. So the way that you do a test cross is you take an individual that has a dominant phenotype, but you're not sure of their genotype, right? That's her. Okay. And you cross her with an individual who is homozygous recessive. Why would we do that? Well, let's walk through it. Okay. Let's go through it. So essentially, this is showing you the two possible outcomes depending on if she is homozygous dominant or if she is heterozygous. Okay. But actually, rather than just looking at the results, let's go through it and actually set up the Punnett squares and do it. Okay. So that we can, have a little practice. All right. So let's go to a white screen here. Okay. So remember, we have right. So like I said before, when I'm writing out my work for a genetics problem, I always try to keep track of everything. Okay. So I know for this particular trait that big B is black, little b is chocolate. Okay. And I know we have a black dog of unknown genotype. Her phenotype is known. right? Because remember, phenotype is what you see. So we can see she's black, okay? Unknown genotype. So what that means is she could be this, or she could be that, okay? So we're going to cross her with a male who is homozygous recessive. So in order to do a test cross, you're always taking the unknown dominant phenotype situation and crossing them with homozygous recessive. Okay. So to set up our Punnett square, we're going to do 2 Punnett squares, right? 1 Punnett square for if she's this and 1 Punnett square for if she's that. Okay. So remember when we set up our Punnett squares, what are we putting on the edges? So what are we writing on the top? What are we writing on the side? So your instinct is just going to be, oh, I'm writing letters, right? And you just want to write the letters. But remember, what do those represent? Well, they represent what are the possible gametes, right? That individual can produce. Okay. So if we're doing our first Punnett square, can't draw a square on a computer to save my life. If we're doing our first one where we're assuming she's homozygous dominant, right? What can she put? What can she put? What kind of alleles can she put in her eggs? Well, all she has are big B's. Okay, so I mean, we could write big B twice, but I mean that would essentially be redundant. So we don't need to. Okay. And then remember that we're going to cross her with our male that is homozygous recessive. So what can he put in his sperm? Well, he can only put little b's. Okay. So do I need to write little b twice? Well, he can only put one thing in his gametes because all he has are little b's. He can only put a little b. Right? So in reality, we don't need these two at all. Okay. And I'll show you why. I'm going to prove it to you. Okay. So big B from mama, little b from daddy. Oh, look, this is just 4 squares that are all saying the exact same thing. So it's not necessary. Now, if it is easier for you to just do a 4 square Punnett square all the time, that's absolutely fine. Okay. But the point I'm trying to make is that, okay, in reality, if all she can give is B and all he can get big B and all he can give is little b, that's all the Punnett square you need, because 100% of their offspring are going to be, okay, all babies will be that, which is black. Okay. All right. Okay. What's the other possibility, right? So that's this first possibility. And then here's our second possibility. So our second possibility, so 1, 2. What's our second possibility? Our second possibility is that she is heterozygous. So her eggs could have a big B or a little b. And then once again, he still can only give one. He can only give little b's because that's all he has, right? So, I mean, you could write little b twice. You could certainly do this, right? Let me figure out how I want to do this, right? We could certainly do another little b over here. I'm going to erase it in a minute, though, because we don't need it. Okay, so let's fill it in across and down, right? So big B, little b, little b, little b, big B, little b, little b, little b. Right? And so remember, this column is just a duplicate of this. So we can actually just do that. Okay. So what does this Punnett square tell us? Well, it tells us that on average, so the probability of having half of the babies will be this, right? And then half will be this. So half will be black. And what does 2 little b's mean? Half of the babies will be chocolate. Okay, so how do we, practically speaking, how do we use this? Okay, so let's say we have our black female and we know she's black. So we know she has one big B because she has a black coat. Okay, but we don't know if she's heterozygous or homozygous dominant. We can't tell by looking at her. So we decide, okay, we're going to do a test cross. So we cross her with a chocolate male because he is homozygous recessive because he's chocolate, remember, right? One of these two things is going to happen. Okay, let's say that they have a very successful breeding and they have a litter of 8 puppies or 10 puppies. I don't care, right? They have a big litter. Okay, and every single one of those puppies turns out to be black. You probably feel pretty confident that she is that. Now, remember, these are probabilities, not promises. So it is possible that she's actually heterozygous, and for whatever reason, you just got 10 puppies like this and no puppies like that. So it's possible, but it's not likely if we're talking about a litter of 8 puppies or 10 puppies. If they're all black, we probably can feel pretty confident that she is homozygous dominant. Okay. Now let's say that we, cross them and maybe the litter isn't as big this time. Maybe they only have 3 puppies, but one of the puppies turns out to be chocolate, right? That's an automatic giveaway that she is in fact heterozygous, because the only way to get chocolate puppies is if mom is at least heterozygous. Yeah? Okay. Or they could have 10 puppies and 5 of them might be chocolate, 5 of them might be black, or 3 of them might be black and 7 of them might be chocolate. Remember, these are probabilities, not promises, right? So it's just like on average. Think about it that way. Okay. But if there are any chocolate puppies at all, you automatically know that she was heterozygous. If there are not any chocolate puppies, now, let's say you bred them together like 3 times, and every time you bred them together, all of the babies were black. At that point, you're probably feeling pretty confident that she is in fact homozygous dominant. You might not feel that confident about it after just one litter though, because it might be, well, maybe we just, didn't get that combination. I don't know. Okay, so that's a test cross. All right. And so, take a mental picture of this really quickly, and then let's go back to here. So because you are used to doing a 4-square Punnett square, that's what they showed you, right? But my point was, we don't need this extra column if these are the same. We just don't need this column, right? And if these are the same and these are the same, we don't need this column or this column because it's just redundant. It just tells you the same thing. Okay. All right. So that's how to do a test cross. So Mendel could do test crosses, right? This was another thing that was on Mendel's, he could do these. Okay. Now we're moving on to things that were sort of beyond what Mendel was able to figure out. Because remember, he was limited. He didn't understand the mechanisms of how all of these things were happening. He just understood that they were happening. Okay. All right. So this is just a little reminder, okay, that dominant and recessive dominant doesn't mean more common or normal. Notice normal is in air quote in quotes. I don't like the word normal. We'll get back to that in a second. So dominant doesn't necessarily mean that something is more common. It simply just means what? Do you have a good definition in your head of what does it mean if an allele is dominant as opposed to an allele that is recessive? What does that mean? Right? So a dominant allele is one, right, that is expressed in a heterozygous individual, right? Recessive alleles can be covered up by dominant alleles in a heterozygous individual. Okay. So dominant doesn't necessarily mean more common. Dominant doesn't necessarily mean better either. Sometimes I think people think like, oh, dominant is better, right? It's not necessarily better. It's just expressed in the heterozygous. Okay. And so here are some examples of some traits that impact humans, right? So we talked about freckles already. Having freckles is a dominant trait. That doesn't necessarily mean that it's more common, right? So In genetics, we call that the wild type trait is the more common, and the less common is the mutant trait. Those when we use those terms for animals or plants, it's, people get sort of like weird about, using those terms for humans because I don't know, they seem judgy. Um, right? So mutant just means it's the less common trait. Okay. So in the case of freckles, even though freckles are a dominant trait, they're the mutant trait because they're less common. Okay. Not as many people have freckles, right? The majority of people do not have freckles, have the recessive. Most of us are homozygous recessive for the freckle gene. Okay. Albinism. Albinism is a condition where it's actually more complicated than they want it to sound in your book, but there's various different types. But in general, albinism is when somebody's body doesn't produce the normal pigment. So you don't make pigment in your skin or your hair or the irises of your eyes in some cases. And so albinism is fairly rare condition. It also happens to be recessive, right? So most of us have, quote, normal pigmentation, meaning we produce pigment. Now, what color those pigments are varies tremendously, right? But, right, it's the difference between making pigment at all and not making pigment. Okay, so albinism is the recessive condition. There are a lot of genetic disorders, genetic diseases that we're going to talk about, a few here and there. One of them is cystic fibrosis. Cystic fibrosis is a disorder that affects your lungs, and it also affects your digestive system as well. And basically what happens in people who have cystic fibrosis is they produce, I'm kind of oversimplifying it, okay, but generally they produce too much mucus. Okay. And so this is a genetic condition, right? It doesn't have anything to do with your lifestyle or anything else. It's inherited in a pretty straightforward Mendelian fashion. It is not super common. So it is a mutant trait and it is a recessive trait. So once again, the vast majority of us, right, are either homozygous dominant, meaning we don't have cystic fibrosis, or at least heterozygous. The only people who have this particular disease are people that are homozygous recessive. Okay. One interesting thing about many genetic conditions is historically, they are more common in certain ethnic groups than others, just by virtue of the fact that for much of human history, okay, not as much recently, but for much of human history, people were separated and it was hard to travel all around the world, right? You had to take a boat and you could be on that boat for 5 years. Okay. So now there's a lot more sort of intermingling than there once was, but even still, some people, many people, right, spend their whole life living in a relatively small community. Okay. So there's more intermingling than there, was ever has been before. But a lot of these genetic disorders are more common in certain ethnic groups just because of their separation. So cystic fibrosis happens to be a disorder that's much more common in people of white European descent.